Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, RedShiftBio, I'd like to welcome you to Conformation Change of a Monoclonal Antibody in Real Time, Mimicking Low pH Hold Viral Inactivation. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now I'd like to introduce our presenters for today. First is Eugene Ma, Chief Commercial Officer for RedShiftBio. And our second presenter is Steven LaBrenz, Scientific Director at Janssen R&D PDMS. Welcome, Eugene. The presenter ball is yours.
Thank you. This is Eugene Ma with RedShiftBio, and I'm going to provide a quick introduction to the AQS3 Pro, which is a new type of instrument that is used for biophysical characterization of proteins. This is the instrument that Steve used in his experiment, which you'll hear about in the second part of this webinar. So as I mentioned, the AQS3 Pro is a new type of instrument that is being used now for biophysical characterization of proteins. Many tools currently exist that cover different types of measurement, like CD and FTIR for structure, DSC for stability, CC for aggregation, and V for quantitation. But industry is never one to sit still and demanding the ability to measure over wider concentration ranges, to have greater sensitivity to see smaller and smaller changes, the extraction of better and more usable or actionable data with each measurement. All this in addition to demand for simplifying the workflow, not just in prep and test, but also in analysis. So we developed the AQS3 Pro to address these needs. It performs 5 key measurements in a single analysis. You see some of them here on the right: aggregation, quantitation, stability, similarity, and structure. It performs this with 30 times the improvement in sensitivity when compared to conventional IR methods. It has the widest concentration range of any platform. We're routinely able to measure between 0.1 to over 200 mg per mil. And it has a integrated multi-sample capability using a well plate design for up to 20 times savings in direct labor costs. And finally, it has an automated and integrated protein analytics software suite, which is able to provide all level of detail, satisfying the needs of operators through scientists. The AQS3 works in the mid-IR, where it probes the vibrational modes of the carbonyl bond in the protein backbone. And this reveals spectral signatures, which are mapped to the secondary structure of proteins. Some of those are shown on the bottom right here: alpha helical structures, beta sheet, for example. And this technique is very powerful. It's been long recognized as a very effective method for the analysis of protein structure. But it hasn't been fully exploited because of constraints in the conventional IR methods available to date, particularly for aqueous solutions. So we developed a new platform, a new technique called Microfluidic Modulation Spectroscopy, or MMS for short. And it's really built upon 3 key components, which are shown on the left: a tunable infrared laser, microfluidic referencing is performed with a fluid cell, and the third piece is our advanced analytics. And so these 3 are integrated together onto a platform which basically works as follows. So on the right here, you see a schematic that basically shows laser absorbance spectroscopy. Our source here is our tunable laser instead of, say, a glow bar, so it's extremely bright. It allows us to use a much simpler detector, a T-cooled detector, and in between we have our fluid cell. Now this cell is actually designed with 2 inputs, one for a reference fluid and one for a sample fluid. And these fluids actually modulate back and forth, typically between 1 and 5 times a second. And this is performed in order to achieve background subtraction. So we're able to do this very, very quickly and very timely. And this is possible because of the geometries of our microfluidic cell. And all of this is choreographed with our integrated software. So it coordinates the activities of our tunable laser with the modulation of the fluids and is able to construct a very accurate and very repeatable spectral profile for all our measurements. So all those put together allow us to achieve a sensitivity which is about 30 times better than conventional methods, all while simplifying the actual operation. We've tried to take the user out of the measurement as much as possible here so that measurements are very, very consistent and repeatable. So to test the performance benefits, we began by first modeling the performance of this platform, comparing it to traditional IR methods. So what you see here is a performance plot with sensitivity on the y-axis shown in milligrams per mil of protein concentration versus path length, cell path length on the x-axis. Shown in the blue and the black are the performance curves of conventional IR tools. You can see that optimal performance there is achieved around 6 or 7 microns path length, which is why if you buy a cell today, they're typically at that geometry. MMS, in comparison, is shown in the red curve here, and what you'll notice is that there's about a 30 times improvement in sensitivity in addition to a much longer path length for the cell. So we go from 6 or 7 microns to typically about 25 microns in path length. We're able to do this because we have so much more optical power from our laser, and this increased path length really improves our sensitivity to conformation chains. So in addition to that, I mentioned that we utilize microfluidic referencing, so we're able to perform background subtraction very, very rapidly and repeatedly. We're able to optimize the optical power that is on our detector because we have a tunable laser here, so we're much more efficient with the dynamic range of our detector. And water vapor correction and subtraction is performed several times a second and is very, very accurate. All this capability really addresses the need for an expanded concentration range. Groups that we've worked with have cited this as one of their major motivations, and one of the key features of MMS that they like. They're able now to measure samples from 0.1 mg per mil to an excess of 200 mg per mil, all with the same tool. So now you can look at data from discovery through formulation with the same apparatus. So you now can measure at the concentration of interest. There's no need to dilute your sample. This simplifies prep and workflow. But more importantly, you're able to actually measure the protein conformation in the formulation of interest, and that's just better science. So these performance advantages were independently validated by Elion, which is an analytics group out in Colorado. And they performed an experiment where they mimicked or simulated protein conformation change by spiking a beta-rich protein, in this case an IgG, with alpha-rich protein, BSA. And they compared MMS, which is shown here on the right, with traditional FTIR, looking at the limit of quantitation using their metric, which is a weighted spectral difference. So what you see on top here are second derivative spectra for the samples. These are multiple spectra, and you can see they overlap fairly well in most places, and the change by spiking with an alpha-rich protein is shown here in this spectral region. Again, FTIR on the left and MMS on the right. When they dive into the data, they see that the limit of quantitation under FTIR measurements here, the concentration I should mention is 20 mg per mil. is about 22.7%. In contrast, MMS yields a limit of quantitation much, much better. It's down to below 1%. So, in fact, this matches up very, very well with our expectation that we'd get about 30 times improvement in sensitivity. What they also noted was that there were significant labor savings. The average labor time for each sample measured under conventional methods was 30 minutes per sample. And with MMS, it was about 1.5 minutes per sample. And this was, as I mentioned earlier, not by accident. This is by design. When we created the AQS3 Pro, we designed it to work with modern workflows. So we integrated a well plate design with it. And this slide just provides an image showing what our typical setup or workflow looks like. Samples are loaded directly into the well plates. That well plate is loaded into our instrument. The user is able to select build or select a preset test protocol, press go, walk away. All measurements are performed automatically here. And you're able to view this at your computer remotely or at the bench. So this is typically what's done or how we use the tool with our collaborators like Steve. And at this point, I guess I will pass the mic over to him.
Good morning. Thank you very much. This is Steve LaBrenz from Janssen. Welcome to the portion of my talk where I'm going to discuss our work around a proof of concept to develop the process to simulate viral inactivation hold and looking at molecule conformation under those conditions as part of a purification development process. So we were looking at conditions where previously in the history of working in this area, we've had some non-conformance events in manufacturing. This is many years ago. And it was the closing of those non-conformance events took months to complete, to plan, to do the work, and to close out. These non-conformances can be purposeful or accidental, as part of development activities. And we deploy standard testing, typically chromatography or mass spec, and over again, there was no indication of risk to the product. We did have a separate technique which allowed us to look across the number of batches involved in those nonconformants and actually see differences across those. And what that led us to do is identify that, once you alter a sample out of that state, that condition where you're mimicking, you're inferring results to the previous state. And sometimes changing the sample can mask those results, and your traditional techniques may not pick up what's going on. So the purpose here was to perform a proof of concept assessment and look at protein stability at low pH over time in reference to the previous work that was done. To discuss what the thought process is behind the experiment, we go back to a combination of colloidal, biophysical, chemical stability that was discussed in Jan Radford's paper in 2005. And I use this figure from their paper that looks at the energy state and across the whole range of protein conformations that can be accessed within a given energy state for a molecule, different buffering conditions. And what we're looking for is ways to assess the conformation where other techniques won't give you that information in this space. And as indicated by the arrow, which indicates a native state or a preferred condition, it's not necessarily the most stable. That is, it doesn't have the lowest energy state across this whole funnel diagram. And we're really interested in understanding how the conformation changes in these different areas in relation to possibly seeing something like an aggregation event, which is shown on the right side of the figure, as well as possibly are there intermediates, folding intermediates that might be leading to that state, or are we picking up changes in these partially folding states? So we want to look across this and be able to identify where our system is breaking down, or if those changes are even relevant to the process. To go over the experiment shown here is a lot of text. It outlines exactly how we prepared the experiment. In brief, MAB was buffer exchanged using the GE Healthcare PD10 columns to obtain exact buffer match between the background and the sample itself. We did some work before showing that this is a great way to generate the background match to the sample. We also then worked together with Redshift and determined how much acetic acid was required to add to MAB in an acetic acid tris buffer at pH 7 to obtain two pHs, pH 3.5 and pH 5. And with that in hand, we are able to generate samples, a large bulk sample, which was then dispensed across the different sample positions in the plate for the AQS3 Pro, and assess those over time. What I'm showing here is the absolute absorbance data, the raw data that is, from the instrument. In the upper left-hand panel, you see samples from 15 minutes to 3.5 hours that were at pH 3.5. The data looks very, very consistent across that approximate 3¼ hour time frame. We then add the control data, the pH 7 sample, to that, which is the upper right-hand image, and you can definitely see in the raw data here a change in the spectral data, indicating a shift in conformation, especially in the amide region. Then in the bottom left-hand corner, what we see is an overnight sample. So the instrument was put into a pause mode or a wait mode, and then in the beginning of the day and the next day, the last sample data was collected. And you see the pink line with a large change again in spectral data. And there's many items that might be represented by that change. It could be a drift in sample change, like background subtraction change, but we believe from including the data and discussion further that this may actually be a true change in conformation in the protein secondary structure. To further process the data, you then take that raw data, perform a second derivative calculation to obtain the second derivative spectra, which is what people are generally used to seeing in publications, because under these conditions, you can then deconvolute this data and understand what different structural components are in your sample. And under here, what you can see again is that you have all three previously identified species, the control at pH 7, the overnight sample, which is in the pink, and then our data from 15 minutes to 3.5 hours under this, what looks to be a red line there. And you can definitely see changes in intensity in the second derivative spectra of the samples. And these will be then deconvoluted to show you different conformational proportions indicated to beta, alpha, turn, and helix. So you can see here the pink line, which was from the control, or I'm sorry, the greenish line, which was from the control. Instead of being very, very different from the previous spectra, which you saw on the last slide, it's actually quite aligned with the other components in here, albeit different. This is another step in the analysis of data, and the delta plots here are a subtraction of the control from all the other spectra, and you can designate which sample you want to subtract from everything as the baseline sample. The control is bracketed by the dashed lines showing the variability of the control, showing that it is in a good, stable sample. And then what we see in this data is a significant difference between the samples from 15 minutes to 3.5 hours, as well as the overnight sample. But we particularly want to examine the time data. And what you see here is an indication at 1638 inverse centimeters, this key signal for a beta sheet structure. You do see some changes that take place over time with this. This data is deconvoluted, or excuse me, first we're going to look at the time chart. And what you see here is the intensities of those signals. And we're particularly looking at the 1638 signal here. And what you do see is you start to see the changes occur once you go past approximately 90 minutes in the data. And what we're indicating here is that this is a conformational change over time at pH 3.5. To first glance here, this may not look like a significant change, but the scale on this plot is only approximately 0.02 units. And we're seeing about a 0.001 to 0.002 shift in the intensity. So we're looking at a 5 to 10% shift in that signal. It may not look like much, but it actually is quite a bit. When we deconvolute the data is where some of our information comes in. In the left-hand side, what we're plotting is the percent higher ordered structure for each of the individual components for a monoclonal antibody. Obviously, we have quite a bit of beta turn as well as beta sheet structure as well as turn structure, and we're low in unordered and alpha structure. What you definitely see here are changes between the beta and the turn significantly from the red, our control sample, to all of our time samples here. It's the overnight sample that then goes back and shows you an increase of the beta sheet structure and a decrease in the turn structure over this period of time. And this is further giving us insight that we believe what we're seeing here is actually not something to do with a long-term hold, but is actually a conformation or a possible colloidal interaction taking place in solution. All samples at the end of the experiment were clear and clean to the naked eye, so there was no obvious precipitation of sample over time. Likewise, when we look at the standard deviation on the data for each of the structures, it shows that the individual numbers do not overlap. Therefore, we believe that the changes are real and statistically different. So as I mentioned, we also ran pH 5, and this experiment was performed in the same way, that the acetic acid was added to the sample, and when in bulk and the sample distributed across the plate. And this experiment shows that, again, over time, there wasn't a huge change in the structure of all the samples. We know for this particular MAB, its preferred pH is approximately 6, which is demonstrated the pH 7 as our control is where we like to keep it around, and that at lower pHs, it has a little bit less stability. But here you can see under these conditions that the lines were pretty flat. We did see this overnight shift in, again, that 1638 beta sheet signal. When we deconvolute the data, and what we have here is a higher ordered structure analysis of this data, you see a very, very small conformational change. And again, when we come back to the numbers, we do see significance. The changes are real, but these differences are very much smaller than what we're seeing in the pH 3.5. As I mentioned, the pH that this MAB prefers is pH 6, And so with less perturbation, that is only going to pH 5, we don't expect as large of changes. So what are we looking for here? So under native sample conditions, that is, under conditions that would be representative of low pH hold in a purification process, over 3.5 hours at pH 3.5, there was a conformational drift. And at pH 5, there was no conformational drift, or very little drift. And so what we're looking for here are conditions that tell us what might be going on in solution that is representative for purification. Why is this important? It's important because it helps us understand the time that the MABs, how they behave over time at low pH for viral inactivation. Our process is typically 90 minutes. And as this data shows, 90 minutes seems to be pretty good under these extreme conditions that we were mimicking here with a lot of additional tris in the buffering condition. And this also gives credence to the ability that even if you're doing very early development, as Eugene was talking about in his slides at the beginning, there's a potential here to use this to find resistance to conformational change. Say you have 4, 5, 6 MABs and you're trying to determine which one to carry forward. This will give you insight into developability. That is, which of the MABs would be most stable against these pH denaturing conditions or pH conformational change. And you could use that as a selection criteria. So why should we use the AQS3 Pro? Well, we've actually followed up this experiment over the previous few months here of the year. And new data from orthogonal assay indicates that this experiment can be improved. As I mentioned at the very beginning, the samples were prepared in acetate tris, which is a normal buffer for this part of the purification process. But what we were able to do is look at samples that came straight off of a protein A column, only in the eluting acetic acid buffer, and determine ways to look at that stability using this orthogonal assay and test different additives. We need to determine, we saw significant changes in that screening, but we need to determine if those observations are related to conformational change, and are we seeing similar or different changes in the intensity of those particular wavelengths we saw under this experiment. Our basic assessment that we've been doing cannot delineate these conformational effects from colloidal effects. With an increased amount of experimentation and observation using the AQS3 Pro, we think that we can answer this question, conformation versus colloidal effect. With the direct sample analysis, as I mentioned, we did this years ago, but we didn't have any secondary structure analysis. We've actually determined, compared to especially a circular dichroism instrument that we have, that our sample testing time was reduced by 80%. That is, we could essentially do the work of a week in a day using the AQS3 Pro. Also, the samples are analyzed under these native conditions, and that gives us 100% relevance to our purification process development. So we can take samples directly from a small protein A elution, and dispense those samples across a plate and put it immediately into the AQS3 Pro. That would allow us to perform the work essentially right there on the spot, relevant to our purification process. With that, I'd like to thank you for your attention and open the floor up to questions.
Thank you very much, Steven, and for your presentation as well, Eugene. So our first question, how much optimization of parameters is needed before measuring each sample or spectrum and for each sample set?
In my experience, it only took us about a morning's worth of work on individual samples to find some optimization conditions. After that, it's generally sound for the molecule that you're examining. If you change the concentration, then there's a few parameters that you might have to quickly adjust, and you can usually do a best guess to make sure that you're collecting good data.
Yeah, Eugene here, I'll just quickly chime in too. If the question is more about optimization of operational parameters based on, say, changes in viscosity, concentration, things like that, the instrument itself has an automatic discovery routine or process that it executes where it will change and vary the back pressure and determine what the most appropriate settings are for the sample that you happen to be testing. And that routine, as mentioned, is automatic and typically runs very quickly, usually in about 2 minutes or so.
Excellent, thank you. Our next question, is there any water vapor subtraction applied to the MMS spectrum?
Eugene, again, I'll take that one. Yes, MMS, as I mentioned in my part of the talk, performs automatic background subtraction, typically between 1 and 5 hertz. So it's essentially a comparator tool, and so any vapor that's present in or during the measurement of your reference fluid is also presumably present when you're measuring your sample a half a second or shorter after. And so those get background subtracted out automatically.
All right, thank you. The next question, what are the upper and lower protein concentration limits for this technology?
Eugene, again, I'll answer that. We routinely measure between 0.1 mg per mil to, as I mentioned earlier, we've gone as high as 230 mg per mil. Most of our testing is done closer to between 1 and 10, but we've certainly pushed those limits. I suppose we could go higher than 230, but we haven't tried yet.
Excellent. Our next question, are any buffers typically found in protein formulations that are incompatible with microfluidic modulation spectroscopy?
We haven't found it yet. We've worked with a variety, a large number of collaborators and looked at many different types of buffers that are typically used in development. We haven't yet come across anything that has been incompatible. Part of the advantage of this technique is that it's just a straight up differential absorbance measurement tool. So if you're able to match your buffer, your reference to your sample, having common buffer there, it will do an excellent job subtracting it out. So spectrally speaking, there's no contamination or interference from excipients. And we haven't come across any buffers that have been problematic from an instrumentation point of view yet.
All right, a very practical question. Do you demo your system?
Yes, absolutely.
All right, our next question from Korth. Why was acetic acid used for pH adjustment instead of a strong acid like hydrochloric acid? It would seem like acetic acid would result in a change in OAC concentration as well as provide an oxidizing agent.
Sure, I'll answer that. Acetic acid is used throughout our purification process, not hydrochloric acid. So that's why it was chosen. I did see the question in reference to acetic acid as an oxidizer. It's our experience that acetic acid actually doesn't lead to these oxidation events. In fact, using hydrochloric acid in biomanufacturing would pit the steel, which would lead to oxidation events.
Excellent. We have a question from Lou. Have you ever looked if the conformation changes reversed during the neutralization step?
Very good question. That's actually the next step after this proof of concept study. So we're expecting delivery of our AQS Pro 3, our AQS 3 Pro this month, and those will be the first experiments going on.
So I guess I guess that answer is a stay tuned for future results.
Exactly. And it's also in combination, as I mentioned, we have more results going on. My best estimation is that we've probably enough new data looking at this low pH hold and neutralization step to provide us anywhere from 4 to 8 months worth of work on the system when it comes to our lab.
Our next question, has RedShiftBio done any work comparing the precision and accuracy of the AQS3 Pro to a CD Spectra device?
Yeah, Eugene here. Yes, the answer to that question is we have. In fact, as part of our work with Elion, which I presented in one of my slides, they also looked at performance of CD. So if you go to our website, you should be able to find some information on that as well.
All right, and we had a follow-up question to the previous question. They're asking, how will you be letting folks know about the results once you get the new unit tested?
I'm guessing that might be for myself. It's possibly stay tuned. We have direct interaction with Redshift regularly, and we might be able to put together a white paper as well as a potential to present at conferences.
Our next question, could you see using this technology to monitor your process at bioproduction scale?
So I believe you could be doing that as long as you classified it as an in-process control test, not as anything that would be GXP reportable, because of that it's essentially you're not at the end of either part of the process. You're not pre-purification, you're not post-purification. That's my opinion. The beauty here is that you could do, as is often can be done in manufacturing, you could be pulling thieving samples from certain points in the purification process. And then because of the short period of time it takes to analyze the samples, you could actually generate the data before the next step is ready to go in the plant.
All right. And Steve, since you're on, we have a question specific to one of your slides. The delta graph that you presented, you can see, well, I'm not sure what the questioner is asking, but differences. Can you see differences in 15 minutes at low pH?
So from our pH 7 sample to the pH 3.5 sample, yes, those differences were there. If we refer back to the delta graph, the first 2 points on the delta graph were actually the control at pH 7. So then the severe drop we saw was the pH 3.5 sample, and that first 3.5 was at 14 minutes. So the control samples were run before that 14-minute sample, and you see that distinct conformational change at that time.
All right, understood. And then Andrea asks, can you calculate melting temperatures?
Yeah, yes. In fact, we've done a lot of thermal stress work. So I would say technically we wouldn't calculate or determine a melt temperature. We do measure and observe a TM where the M might be a midpoint temperature. Basically what's done is we are able to thermally stress proteins outside of the instrument for now, quench it and look at the conformation that way. And when we plot a spectral change at a particular signature point against, say, the stress temperature, we get a plot that looks very similar to what you might see for a DSC. And from that, you can calculate a midpoint temperature.
Ah, okay. And then, Eugene, a question for you from Prothema. What is the requirement for the purity of samples for this measurement and interference of HCP?
So I guess, yes, it ultimately depends on the application. Again, when you dial everything back, the instrument itself is a comparator. So if you are able to match impurities that might be in your sample also in your buffer, then it's not an issue. Otherwise, you will, like with any spectroscopic method, be measuring the difference. In this case, it might include the impurities. So if you're able, like excipients, various excipients that might be in your buffer, if those are captured in your reference, then it's not an issue.
All right, and a follow-on question to that one. Has this been tried for the VI of other recombinant and fusion proteins that don't use protein A?
So we have worked with a number of collaborators looking at various proteins. There are a lot of MABs, but we have looked at fusion proteins, ADCs, peptides as well.
Yep.
And another VI question for you. Lou asks or says, generally as part of the VI studies, we apply mixing at a defined mixing rate. Is this capability possible within the cuvette during measuring?
That's an interesting question. It's something that has come up in discussions we've had with various groups. So in principle, yes, it's one could envision doing that. The tool right now and the cell that we've designed for it is not designed to perform the mixing, but one could see that with some clever designs, those types of applications could be addressed with this tool.
Excellent. And then a final question from Michael. Have you had the chance to correlate the conformational changes seen and the formation of multimers or aggregates in subsequent DSP unit observations?
Yes, we have. That is work that we did with an academic group. It's not yet published, so I can't yet comment on it, but we have matched those up, yes.
All right, that appears to be all of our questions for today. With that, I'd like to thank so much our presenters for today, Steven LaBrenz and Eugene Ma. I'd like to thank RedShiftBio for sponsoring today's web symposium. And most of all, I'd like to thank those of you who came and spent this time with us. We hope you gained some answers that will make your life in the lab a little bit easier going forward. So on behalf of Cambridge Health Tech Institute's Global Web Symposia Series, thank you all so much and have a great day.